Hello everyone, my team and I are going to be explaining how to perform the polyacrylamide gel electrophoresis and the agarose gel electrophoresis. And I hope that after this explanation that you will not find it difficult in performing these two powerful laboratory techniques. Thank you.
Okay, here are the materials used to perform agarose gel electrophoresis in the lab. The materials are agarose powders, electrophoresis buffer, example TAE, DNA samples, loading dye, DNA size, DNA ladder, that's size marker, ethidium bromide, casting gel tray, comb, and electrophoresis chamber, power supply, and UV translators. What is agarose gel electrophoresis? Agarose gel electrophoresis is a method that uses a gel-like material called agarose to separate DNA fragments. DNA is negatively charged. So when an electric current is applied, it moves through the gel towards the positive side. Smaller pieces of DNA move faster, while larger ones move more slowly. The difference in speed allows us to visualize and compare DNA sizes. Methods in preparing agarose gel electrophoresis are: number one, preparing the gel. To begin, we prepare an agarose solution and pour it into a gel tray. A comb is placed at one end to make small wells. Once the gel cools and becomes firm, those wells act as pockets where the DNA sample will be loaded. The gel is then placed inside a tank containing a salt-based bonding buffer. This buffer helps the electric current pass through the gel smoothly. Number two, loading the DNA sample. After setting up the gel, we gently load our DNA samples into the wells. Each sample is usually mixed with a colorful loading dye, so we can see it easily tracked how far it travels during the run. We also load a DNA ladder which contains DNA fragments of known sizes. This ladder acts as a ruler to compare our sample later. Thank you.
Step 3. Running the gel. Next, we attach the gel tank to the power supply. When the current is turned on, the DNA begins to migrate through the gel. Remember, DNA is negatively charged, so it moves towards the positive electrode. As the run continues, the DNA fragments separate based on their size. Small fragments travel further, and larger ones stay closer to the wells. Step 4. Visualizing the DNA. After the electrophoresis is complete, we take out the gel and place it under a special light source, usually blue or ultraviolet light. Before this, the DNA is stained with a safe dye that makes the bands glow so you can see them clearly. Continuing, what we see is a series of bright bands, each representing DNA fragments of different sizes. By comparing the samples to the DNA ladder, we can estimate the size of the fragments. Step 5. Interpreting the results. Finally, we photograph the gel or analyze it using a gel documentation system. The position of each band tells us how large the DNA fragments are and whether the sample contains the DNA we expect.
I'm going to walk you through how to perform polyacrylamide gel electrophoresis, commonly known as SDS-PH. This is a technique used to separate proteins based on their size. Let's get started. First, we prepare the protein samples. To do this, we mix a protein with a sample, loading buffer that contains SDS, using agents like beta-maker, ethanol, or DTT, glycerol, and a trachytide. The SDS unfolds the proteins and gives it all uniform negative charge, while the reducing agent breaks bonds with sulfide bonds. After mixing, we heat the samples, usually at 95 degrees Celsius, for about 5 minutes to make sure the proteins are fully denatured. Second step, setting up the gel. Next, we take our polyacrylamide gel, which usually has two parts. Firstly, a stacking gel at the top and a resolving gel at the bottom. The stacking gel helps protein give up neatly before entering the resolving gel, where real separation happens. We place the gel into the electrophoresis tank and fill the tank with running buffer, making sure the wells are fully covered. Third step, loading the samples. Once the gel is set, we carefully load our samples into the wells. It's important to use a steady hand and avoid puncturing. the wells. We also load a protein ladder or marker in one of the lanes. The ladder has proteins of known sizes, which will help us estimate the sizes of our sample proteins later. Thank you.
The fourth step, running the gel. Now it's time to start the separation. We connect the tank to a power supply and run the gel at constant voltage, typically between 80 to 120 volts, depending on the gel type. Because the proteins are seated with negative charges from SDS, they will migrate towards the positive electrolytes. Okay. Right. The fourth step, running the gel. Now it's time to start the separation. We connect the tank to the power supply and run the gel at the constant voltage, typically between 80 to 120 volts, depending on the gel type. Because the proteins are coated with negative charges from SDS, they will migrate towards the positive electrode. Similar proteins move faster through the gel pores, while larger proteins move more slowly. The difference in speed creates the separation we are looking for. The fifth step, staining the gel. After the run is complete, we remove the gel and place it into a staining solution, commonly Comasi brilliant blue or sometimes a silver stain. The stain binds to the protein, making the bands visible. We then stain the gel to remove excess dye, leaving only the stained protein band behind. Analyzing the results. Finally, we look